This lecture is going to involve, again, talking about the adaptive immune system, our third line of defense, and the types of cells that are involved. If you recall when we talked about the innate system, there are cells like the dendritic cells, like the macrophages, that also help to communicate with the adaptive immune system. So here we're going to discuss those two types of lymphocytes, the B cells, which are part of the arm of the humoral immunity, the humorals being the fluids, blood, interstitial fluid, lymph, things of that nature, and the T cells, which are involved in the arm of cellular immunity, meaning that they recognize your self cells that might be infected internally or your self cells that could have become cancerous. Then we will discuss the antigen-presenting cells. Again, these like the dendritic cells, the macrophages, and I'm going to floor you next when I say even the B cells are antigen-presenting cells. These guys do not respond to specific antigen as far as when they are presenting. They just munch up bits and pieces of the foreign invader and present them, those antigen pieces, to our lymphocytes, our T cells and our B cells. So they do play a role in the innate system, and they also play a role in activating the adaptive immune system. There are several steps from origin of these cells being born all the way to them being active in an infection. And the steps are broken down into 5 regions. The origin, okay, so they originate in the red bone marrow. We've already covered this when we talked about the blood chapter. They are born in the blood marrow depending on what types of signals they receive. Those progenitor cells will turn into T cells, B cells, macrophages, neutrophils, whatever we need, whatever signals they receive. So we should be pretty comfortable with that concept. Next, they need to mature. We need to make sure that they can do their job. So they need to go to school, the B cells and the T cells, and they do that in 2 different areas. We'll talk about both of those. Then we'll go into, they have graduated, they're going to head out into the world, into your body, and they're going to do what's called seeding, which is they're going to plant into the secondary lymphoid organs. All those lymph nodes, those hundreds of lymph nodes, they're going to get in there on that jungle gym of reticular tissue and start circulating around from one to the other, mobile through the body, hanging out here and there for a while, to potentially come across the antigen that they have been trained and schooled to recognize and destroy. Some of your T cells and B cells may never be activated, but if they are, we're going to get into step 4, antigen encounter and activation. How does this occur? What are the steps? And then into the proliferation. Basically, they're going to clone themselves out like Star Wars, the Clone Wars. They all look the same. We're going to make a huge army that recognize that one specific antigen and knock it out. And part of that proliferation process is going to create a nice little subset of memory cells. You can kind of consider those maybe the generals. They are going to hang around forever, and if we ever come across that specific antigen again, they are going to be able to divide faster. They're going to be able to attack faster in a quicker timeline. Because of that, there's less steps involved from those memory cells becoming fully activated compared to this process here where it's the first time they're seeing antigen. They're naive cells. They haven't done the job yet, so it takes them just a little bit longer to gear up. So let's work through these 5 general steps. Great image in the textbook here for you, walking you through both B cell and T cell proliferation. So you can see that we have our precursors in the bone marrow that are going to turn into specific lymphocytes. All right, either T cells, which are then going to move on, or B cells, which are then going to have some more training in the bone marrow. So this is the origin component. Both B cells and T cells originate, they're born in the bone marrow. Second step was that maturation process, all right, going to school, making sure they get their diploma for the one antigen they need to recognize. This happens in 2 different locations. So those precursors are destined to become T cells. They are actually going to migrate out of the bone marrow into the blood and finally end up in the thymus. All right, T cell thymus. This is college for the T cells. This is where they're going to get a couple different tests, positive selection, negative selection, which I'll go over. And if they graduate out into the environment, For the B cells, they get their schooling and training, the tests they need to pass in the bone marrow itself. And this process, which I'll go over next, of positive and negative selection, is going to create mature lymphocytes. So they are going to be immunocompetent. They'll be competent to function in the immune system. Or if they don't pass the tests, they are going to be told to undergo apoptosis and die. If they're not going to cut the mustard, they're gone. For those that do pass the tests, they head from the thymus or the bone marrow, depending on if they're T cells or B cells, into circulation. They're going to go into that seeding process where they are immunocompetent, they can do a job, but they still haven't done it yet, so they're going to be considered naive. And they are going to seed or sit them down into the lymph nodes, into the secondary lymph tissue, the malts. They're going to hang out there and circulate every once in a while through the blood supply until potentially they come across that antigen that they have been trained to recognize. Right? And that is the next step. If they see that antigen, that one little bit and piece that they are supposed to recognize and go after, this is going to cause a process of activation, where either the T cell or the B cell, once it binds to their receptor, it's going to kick off that activation process. And what they're going to do then is go ballistic. They are going to be dividing like crazy, proliferating, creating clones, twins upon twins upon twins, and differentiate into active T cells or active B cells. And we spoke about active B cells making all the antibody. They're called plasma cells. T cells, depending on which type of T cell it is, will have a different name as well if they're activated. Some will be called effector cells, and those are the ones that are going to go out and actively kill self-tissue that's infected or cancerous. Some are going to become memory cells. Some are going to become helper cells. And those are CD4 T cells as opposed to CD8s. There's lots of different kinds. But the big picture is when they see that antigen, they are going to proliferate like crazy, make a ton of effector cells to help fight that infection, because this is level 3. This is that last line of defense. So that's the big picture there, 1 through 5. Let's go into a little more text on those. We know about origin already, covered that. So that's step 1. Step 2 again is that maturation process. This is where the T cells and B cells become educated to find that one antigen that they're trained for. B cells do this in the bone marrow. T cells complete this in the thymus. When they have gone through their tests, again, they are called immunocompetent. Competent to form an immune response. Okay? They can recognize one specific antigen, bind to it, become activated. Now, all your B cells and your T cells display a very unique receptor, again, for that one antigen they're looking for. If they were all looking for the same thing, you'd have a lot of holes in your immune system. So we cover a lot of different types of antigen. Okay? When they bind, they will achieve maturity and bind to that one antigen that they're looking for. We definitely want to make sure that they are self-tolerant, okay? Tolerant to our self-proteins, anything that we make in our genetic makeup. We don't want them to recognize, because if they do, we are going to have an autoimmune disorder. Okay? We want lymphocytes unresponsive to our own antigens. Here is how T cells mature, and then we'll talk about the B cells. T cells, you know, they mature in the thymus, and they're going to take 2 tests. One is positive selection, the next is negative selection. And here's the big picture. They were formed in the bone marrow, they headed to the thymus. The first test, the positive selection, wants to make sure that the T cells are first of all capable of recognizing those MHC proteins that are on the surface of all of our cells. Remember I mentioned that the MHCs are like the hot dog buns, and they'll hold out little bits of protein or components that are in the cell. And if they're self, the T cell, when it binds to it, says, okay, we know that it's supposed to be here, great. And if it's something that it's not supposed to be there and this T cell recognizes, it's going to cause an immune response. So first of all, we got to make sure that the receptor on the T cell can recognize the shape of that hot dog bun and actually fit in a lock and key. And you can see this one did not form properly, whereas this one did. Positive selection here. If it doesn't fit, lock and key, there's some misformation, That is going to cause failure to recognize that MHC, so it's not going to be able to work out in the world of the body, and this cell will undergo apoptosis, basically cell suicide. That T cell is knocked out. If it is able to lock and key fit and recognize a self-MHC molecule, we have passed positive selection, the first test, and then this T cell will go on to the second exam. Okay, the second test is called negative selection. Negative selection, again, if it does not pass, it's going to undergo apoptosis. The T cell is going to die. This test is very important because now we know that it will lock and key fit, but we want to make sure that that T cell does not recognize self-antigens, right? So everything we can make with our genetic makeup, our DNA, we produce that in the thymus, chop it up into little pieces, and put them on presentation cells on MHC. And there's a whole bunch of these cells in the thymus. And then we run the T cells that pass the first test all along these cells, have them shake hands, and if any of them do recognize and would become activated by a self-protein, they are going to undergo apoptosis and die. So this is a way to ensure that we do not create T cells that recognize self, because if we did, again, that is going to roll into autoimmunity. And this process isn't perfect. Sometimes these cells do get through, and that is what causes some of our autoimmune disorders. But if we get through with a T cell receptors at the right shape, and it doesn't recognize any self-antigen, we now have a happy, healthy T cell that is immunocompetent and can be sent out into the world. That is T cell maturation. B cell maturation. Same idea. It is going to go through two tests, both positive and negative selection. It's going to do this in the bone marrow. And again, if it does not pass these tests, it is going to undergo apoptosis, which you can see here on the right. This is a healthy cell, and this is one that's blibbing off, undergoing programmed cell death. So for B cells maturing in the bone marrow, positively selected cells are going to be occurring if they successfully make an antigen receptor, right? Their antibody. So same as the T cells, they got to make an antigen receptor, okay, that antibody that fits. Okay, it's going to be able to recognize antigen. Next, it is going to be going through the negative selection process. If those antibodies that it can produce, which are on the surface as the B cell receptors, if they recognize anything that's self in all those cells in the thymus that are presenting all different bits and pieces of our proteins and would become activated out in the body, again, it is going to be knocked off via apoptosis. So this is a good thing. They can find antigen, but they can't activate against our antigen, both T cells and B cells. So now we have another immunocompetent set of B cells that are going to head out into the world. And the world, of course, is your body. So we have gone through immunocompetence, but they are naive, right? They're just getting out of school. They haven't had any real world experience yet. They are going to head off to lymph nodes, to secondary lymphoid tissues, okay, spleen, things of that nature, and hang out for a while. See if they find or are exposed to antigen that they have been trained to attack. So they're exported to the primary lymphoid organs, bone marrow, thymus, to seed into the secondary lymphoid organs, lymph nodes, spleen, etc. And this will increase the chance of encounter with antigen, because as all that lymph flows through all these lymph nodes, hundreds and hundreds of them, we've got a good shot of coming across antigen that should not be there. All right. A T cell or B cell gets very excited when it makes the proper lock and key fit on the antigen it was trained to find. These are all snipers looking for one particular antigen to kill. This is going to be that last step, antigen encounter and activation for clonal selection. We're going to clone them out, right? Divide, 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 make lots and lots of twins. So those naive lymphocytes, B cell or T cell, encounter the antigen they were trained to see. They are selected for further development. All right, they are going to protect and serve, and start doing the job they were trained to do, which is going to be proliferate like crazy to go attack as a big army and complete the differentiation process from B cell to plasma cell to make antibody, or the T cells into the effector T cells, which are upregulated as well to do their job, killing self-tissue. Step 5, kind of went over this a little bit already, that proliferation and differentiation step. Okay. Once they're activated and start to proliferate, they clone themselves out. Lots and lots of twins, all fighting the same antigen. Most of those cells that divvy out, and I promise you there are more than 7, become the effector cells. These are the ones that are going to fight. Cytotoxic T cells are going to start killing cancer cells or killing virally infected cells. The B cells are going to start spitting out antibody and become plasma cells. However, I mentioned that a small fraction of those cells that divvy out are going to be special. They're going to be memory cells. And what these guys do is they don't fight, but they are left behind after the infection's cleared and all these effector cells die off because they're no longer needed. These guys hang around in the lymphatic system, and they're going to be able to respond if we see that antigen again. They're going to be able to do it faster and quicker because they are no longer naive. They're going to take it by the reins and have a stronger, faster response, more proliferation, shorter timeline to maturity. This is true for both B memory cells and T memory cells. All right, and they will continuously circulate through your body for your lifetime, looking for that antigen again. Summary, in your book there is a table, kind of gives you a breakdown between the B cells and the T cells of that adaptive system. So we know the type of immune response is considered humoral, outside of the cells for B cells, cellular, the cells themselves, for T cells. We know that B cells make antibody and T cells do not. We know the primary targets for B cells is extracellular, right? Bacteria, fungus, parasites, okay, even some virus that's in the extracellular fluid and hasn't entered any of our self-tissue yet. And that's where the T cells come into play. Any intracellular pathogens like virally infected self-cells, like cancer cells, that's who they're going to attack. They're both born in the bone marrow. They go to school in 2 different places, B cells in the bone marrow, T cells in the thymus. They come out as naive cells, and when they are activated, the effector cells are called plasma cells if it's B cells. These guys make a lot of antibody, and they also make a set of memory cells. And for the T lymphocytes, the effector cells have several different names. There's different types of T cells. We can have the cytotoxic T cells, TC cells. These are the ones that are going to be toxic to our cells. These are the killers. We're going to have helper T cells. These guys help to orchestrate the immune response. They talk to several other different types of cells to keep everybody working on the same goal. And another cell type that we're not going to discuss are actually regulatory T cells. These guys can regulate and actually down-regulate a response so that it doesn't get out of hand. All right, but we're going to focus more on the cytotoxic and the helper T cells for future talks. And yes, we get memory cells out of the T cells as well. So there's a good overview of what's just been discussed. Antigen receptor diversity. How in the heck do we get all these B cells with the B cell receptors and T cells with the T cell receptors to recognize so many different antigens? Think about how many different bacteria, how many different components are on those bacterial surfaces, viruses, all the different components on the surface of those to recognize, different types of cancer cells, right? There have to be so many different receptors on T cells and B cells, but we have a very limited amount of genes to create those receptors. How the heck do we do it? And it's all down to your genetics. Your genes are going to determine your immune system's ability to respond. How many receptors can you make? Now, lymphocytes, T cells and B cells, can make up to 1 billion different types of antigen receptors. Out of just 25,000 genes, we do that because we cut them into little pieces, do little snips, use gene segments, shuffle different pieces of these genes together to make different receptors, to make that billion different types. And that process of cutting and shuffling and splicing and dicing those gene segments is called somatic recombination. Now, we are not going to go through this process. It is very labor intensive, but I wanted you to realize how many different antigens your body can respond to with your T cells and B cells from a very limited amount of genome. Somatic recombination is a wonderful thing. All right, we have talked about our lymphocytes. Now let's talk about those antigen-presenting cells. Right? And we already have to some degree. We've talked about dendritic cells, what they can do, munch up and present. We've talked about macrophages as well. These are all types of antigen-presenting cells. And I'm going to blow your mind again. B cells are even antigen-presenting cells. So these cells, the dendritics and macrophages coming off of the innate system, right, non-specific, tie into the adaptive immune system to help activate them. So there is a crossover. They don't respond to specific antigens, but they are very essential in activating adaptive immunity. Antigen-presenting cells. These engulf the antigens, right? Phagocytosis, munch it up into the bits and pieces, the fragments, those antigenic determinants. Okay, and can present those for T cell recognition. This is a lovely little dendritic cell over here. This is electron microscopy. We also have macrophages that can present, and even B cells. More on that in just a second. So here we have our DCs and macrophages again. Just a little overview and a little more information on them. Dendritic cells, they phagocytize the pathogens, right? Munch them up. They then go and enter into the lymphatics, chew up all those pieces, and present out on the surface those little pieces on the MHC molecules. So the T cells can do a handshake and see if they recognize that little piece of antigen. The dendritic cells, as I mentioned, they are more presenters than eaters, big eaters like the macrophages are. So these guys are keyed more towards the presentation. And they are a big link between that innate system and our adaptive immunity of the T cells. Macrophages. Here's a good scan of a macrophage. Widespread in the lymphoid organs. Remember, all of them built into those lymph nodes and the other tissues to help present. They're in connective tissue as well to help munch up anything that gets in there. They can head back to the lymph nodes as well and present antigen to the T cells to activate them. But these guys are more on the big eater side than the dendritic cells are. Dendritic cells are more for presentation. Macrophages even help to secrete antibacterial chemicals to help destroy bacteria without even having to munch them up. Those are two antigen-presenting cells that we know of. Here's the new kicker. B cells, yes, they produce antibodies. So B lymphocytes do not activate naive T cells. I want to share that with you. What they do is present antigens to the helper T cells I mentioned earlier. Okay, and those helper T cells actually assist in activating the B cells. So B cells basically present to the T cell and say, hey mom, is this okay? Is this the one I'm supposed to attack? And the helper T cell will say, yes, that is a great match. Go out and kill my son. This is the process here. B cell finds an antigen that actually fits, lock and key. That one doesn't work, that one doesn't work. Oh, yay, this one fits. The helper T cell will come along and they will discuss via chemicals and contact that yes, you should activate against that. Go on, my child, and proliferate. The B cell will then create a whole bunch of plasma cells. That's all this cloning and proliferation to make antibody. And make a few of those memory cells as well to hang out in your body forever in case that antigen comes back again. All those plasma cells start spitting out antibody, attacking this antigen by binding to those little antigenic determinants on the surface. And then our happy little big eaters, our macrophages, say, yay, I recognize that flag. I'm going to eat you. You look delicious. And those memory cells will hang around, and maybe years later, you're exposed to that same bacteria again, and that memory cell will say, I remember you, and I don't need to ask mom's permission anymore. She already told me once you're bad, so now I am going to create another immune response, but do it faster because it didn't need to ask for help, right? So we eliminate stage 2, and it will clone out faster, make more plasma cells, make more antibodies than it did the first time, and yes, make another set of memory cells to go on if you see that bacteria a 3rd time or 4th time or 5th time. Stop eating those mud pies, people. That is B cell activation. Question time. The adaptive immune system involves 3 major cell types. You have your antigen-presenting cells, the T cells, which constitute blank immunity, and B cells, which govern blank immunity. So take a peek. Which one do you think fits in the first blank? And that'll help you narrow down, and then you can look at the second blank as well and see where those fit. So is it nonspecific and specific? Antigenic, allergic? MHC and MAC attack? Please don't pick that one. Cell-mediated and humoral. You are absolutely correct. Cell-mediated and humoral. Okay, to protect and serve. Lymphocytes are educated within primary lymphoid organs. Right? B cells are educated where, and T cells are educated where. And I know you guys have this down pat, so I'm not even going to wait here. Bone marrow for the B cells. B, bone marrow. T, thymus. T cells go to the thymus. During clonal selection of B cells, those B cells with the complementary membrane receptors to that invading antigen, okay, lock and key fit there, they're going to differentiate into what type of cell. What's the name change when they become activated? And I know you got this one down too, so we will move along. Plasma. The plasma cells are B cells that are spitting out antibody by the tons. Which of the following is not an antigen-presenting cell? I gave you 3, and I think you're going to recognize the one that is not. You are correct. T lymphocytes do not present. Our dendritic cells do. Our macrophages do. Our B cells present to helper T cells to make sure they're seeing what they should and get the thumbs up to produce antibody, but not T cells. All right, summary for this section. Uses lymphocytes, the adaptive immunity, uses antigen-presenting cells as well, and some specific molecules to help identify and destroy non-self substances. Virus, bacteria, parasites, what have you. This is going to depend on the ability of those cells too. They've got to be able to recognize antigen. They've got to be able to bind to it, which is why they all go to school to make sure they can do that. And then they need to be able to communicate with one another so that we get a whole system response. And that ties back into the interferon gamma I was speaking of that helps cells talk to each other. We're also going to see some other components that they talk to each other with little texts and emails. And the T helper cells that I showed you that activate B cells become very important in orchestrating an immune response. An adaptive immune response, I should say. All right, that is the end of this section. Ta-ta for now, and we will come back and do a little more discussion on the immune system.